Hello everyone again, and today we are going to discuss about drug discovery against NIH category A to C pathogens through MEP pathway. MEP is methylerythritol phosphate pathway. I will go through this introduction fast to save some time. The problem is infectious diseases like especially MTB, staph, and enterococci. You can see this 1/3 of world's current population contains latent TB, and we don't know what's going to happen as long as it is people are in safe conditions, And every year, 14.6 million chronic active disease develops, and 8.9 million new cases and 1.6 million deaths every year because of the MTB. Other than this, we have multi-drug resistant TB, extensively drug-resistant TB, HIV TB co-infections, and a lot of terrorism. There is a biothreat agents are also there. These are some of the examples I have given here, but these are the reasons we need to have new drugs. So, especially like for MDR TB, XDR TB, HIV TBs, and drug-resistant, generally drug-resistant gram-positive or gram-negative bacteria. So, what is the current problem with the current technology is fast curing, especially for the TB, you need 6 to 12 months of medication, and it requires combination of several antibiotics, and it easily develops resistance and lots of side effects in it. So to solve all this problem, our lab is doing many different research. I will go through one by one here. The first one is a glyoxylase pathway. So we are trying to determine an inhibitor for this glyoxylase pathway. The mechanism, what we do here is completely different from other inhibitors. So here we induce suicide by the bacteria. So we induce the bacteria to die by itself. But I'm not going to talk about this today. And the other one is the gallium complex. We use a lot of gallium complex as antimicrobial agent. This is a successful program, and we have drugs in our hand, and we have done a lot of preclinical studies, and we want to do clinical studies in this. We are looking for collaborators, especially for the HIV TB co-infected persons. Here, the interesting thing is it's a single drug which can treat both HIV and TB. That is an interesting part of this program. And we are looking for collaborators to do clinical trials for the HIV TB co-infections. This in US here, patients with TB is very difficult to find. That's why we want to have a collaboration abroad who has HIV and TB patients. And the third one is nanopart. This is these two. Most of them are drug discovery type, and here this is a nanoparticle is a drug delivery system here. So we use different kinds of excipients and make conventional drugs, and as well as our new drugs, and put it into a nanoparticle and deliver it to the. In our program, we deliver it to the macrophages. Macrophages are the receptors for both the TB and HIV. So we concentrate there. And the next one is the MEP pathway inhibitor. I'm going to talk about this MEP pathway inhibitor today. And the next one is the MEN-A inhibitor. I spoke about this in the last meeting, actually, so I'm just mentioning it here. So we have a lot of things to talk, but we don't have enough time. And HIV exosomes and drug delivery, that is also undergoing in our lab. We completely studied the exosomes created by the HIV in the macrophages, and we want to use as a drug delivery system in future, but that is future plan. And the last one is we also do the metabolomic studies in neurodegenerative disorders, especially since we do a lot of HIV and bacteria studies. Here we do neuro-HIV infections, that is HIV infection in the brain, and see the metabolic changes in that neurodegenerative disorders here. So let's go back to our MEP pathway. So before, a couple of speakers spoke about isoprenoids here. Isoprenoids is one of the oldest and it's abundant classes of biological compounds known, and both primary metabolites and secondary metabolites are there. And how are they made? So we want to target the synthesis. The MEP is targeting the synthesis of this. So how are they made? There are two different ways of making. I would like to go a little bit faster, so I will just detail what is happening here. So here you can see IspC is used, IspD, IspE, IspF, IspG. These are the proteins, enzymes used to make diphosphates, diphosphate compounds. And this is called MEP pathway, non-mevalonate pathway. And the other pathway is the mevalonate pathway. Here, there is no methylerythritol. Yeah, here there is no methylerythritol phosphate in this mechanism here. So the methylerythritol phosphate is this. Yeah, this guy is the metylerythritol phosphate, MEP we call it here. It's difficult to see for me from here. So this is the MEP. If you see this pathway, there is no MEP in this pathway, but this pathway has MEP. So this pathway and this pathway both produces these compounds. The interesting thing is here, this pathway is only in bacteria and not in human. And this pathway is in humans. So we target this pathway so that it will not give any side effects or infect or make any effect on human. So this is called the MEP pathway. And what it does, see here, it is after the isopentenyldiphosphate, it makes FPPs, UPPs. So what all these things do? So it is used for carrier of sugar for biosynthesis of arabinogalactan, arabinomannan, lipoarabinomannan, and synthesis of dolicol, cholesterol, ubiquinone, menaquinone, lipid 1 and lipid 2, and peptidoglycan synthesis for cell wall. So this is the way in stage 1. You can see forming a lipid 1, and lipid 2 is formed, and peptidoglycan is formed. So this pathway is very important in bacteria to make the cell wall. And as I said, this is the pathway where the IspC is used. First DXS, IspC is used, and then IspD for coupling with the CMP here, and then CDPME IspE to make the phosphorylation at this place here, and then IspF is used for cyclization here, and then IspG and IspH is used to make this diphosphate compounds. So our I'll go here. So here I'm showing some of the examples of pathogens compared to similar to the E. coli here. So Bacillus, Burkholderia, Francisella, Listeria, Mycobacteria, Vibrio, Yersinia, Staph. So most of the pathogens has not more. Every pathogen has this pathway, MEP pathway, and this is similarity to the E. coli. That is why you can see a lot of differences here. These are in very high 80-90%, and here it is 30 to 40%. But all the pathogens has this pathway in it. I would like to show one more thing here. If you see here, this E. coli pathogenic, here the ISPF is not found. You can just keep it in mind. At the end of the meeting, my talk, I will explain what happens. If this is not there, how can it make the compound, you know? This is not the focus of my presentation, but this is one of the findings we observed. So the MEP pathway is attractive target and for developing broad spectrum antibacterials, and it is considered a potential target for antimalarial too, and no reported homologous enzymes in human cells. That is an important part of this pathway. So our group has established this DXS, IspC, IspD, IspE, IspF, and IspH. All these proteins, all the enzymes, we have the plasmids. We have cloned it, expressed it, and purified it, and we are in the process of doing the assay, kinetic assays. of course, we have to find the IC50 for the compounds, so we need the assay and the substrates too. Otherwise, we can't do the IC50s. And of course, we do the MIC too. So we are working on all those things. So to start with kinetic analysis, we need the substrates. So we want to buy this compound. We look for it. It is 5 milligram is $1,000 for this compound. And it is only 50% pure, maybe 60% pure. I don't want to use that one, It's 5 milligram is $1,000 and it's only 60% pure. And how can I find the exact IC50 with that one? So I don't want to use that one. So we decided to synthesize our own compound. Similarly, went for this compound to do the IC50 for targeting IspE. This compound, see, it looks very simple, but 1 milligram is $10,000. This one. 1 milligram is $10,000. Then again, we want to synthesize that one. Again, if you see, the compounds are chiral pure. You can see the the configurations here, and we want the pure compound. Why do we want the pure compound? Of course, I think everybody knows, but just for some of the audiences who are not familiar with the configurations, here if you see the it's a lemon smell and orange smell are different just because of the configurations. And here it's a thalidomide, the poisonous one, and another one is a sedative because of the configurational change. And bitter taste and sweet taste for this, again, because of the configurational change, three-dimensional change. And even it causes the ethambutol as a tuberculosis and it causes blindness, the other configuration. So the pure chiral purity is very important for the drugs. So we decided to synthesize our own compound with enantiomelically pure. This is one step we used, starting with the xylofuranose. We phosphorylated and then oxidized and then protected. This is methyl introduction at this place here, and then benzylation, and then deprotection of this place. Here, oxidative cleavage, and then reduction, and then deprotection using palladium carbon. We got the 100% pure MEP, methylerythritol compound, phosphate compound. And we have plenty, and we do the assays with this one. And the next step, the next compound, which I told 1 milligram is $10,000, we just produce from here with one step with the cytidine triphosphate, and we have hundreds of milligrams of this compound now. This is the first time we synthesized very pure MEP and CDPME of the compound. With this, we want to do the screening. So we had the IspD crystal structure and then did the virtual screening for these compounds. With 20 million compounds, we did the screening with 20 million compounds with IspD inhibitor, and we took the top 10 compounds. And even though it's a virtual screening, we have to test in the bench, otherwise you can't. So we tested it for the IspD compound, but unfortunately none of the compound was a hit in the bench. We thought, anyway, we have these compounds, we have other enzymes, why don't we try in the other one? And accidentally, it was hitting the IspE, the other protein. So even though it is not IspD inhibitor, and we got the IspE inhibitor from the same screening of the compound here. And we did the high throughput screening. Since we have the assay in our hand, we did the high throughput screening in Harvard School, and we got many compounds, the top compound, which is in around 10 to 20 micromole IC50 compounds of this one. So the next step is to go for the IspE inhibitor. Since we have the compound assays and everything, we want to target the IspE. So we did a modeling and found this balanol, this compound balanol is best docking with this IspE protein. So we synthesized this compound. It is unavailable completely, so we have to make the compound. So I'll go faster, just using the caprolactam protection here, and then salination here, and then the dioxidation here, hydroxyl groups, and then protection of the one of the hydroxyl, and then acid substitution at this one place, and then reduction. And we got one part of the balanol compound. Balanol is it's a natural drug, and it is it's very expensive, and it is not available in the market too. So it is very good to make the balanol. Actually, the balanol is also an anti-cancer drug. So the another step, we used the other part of it, but it didn't work, so I'm not talking about it. So we used a different scheme. Here the anhydride is used, ring open, and then protection, and then deprotection of the benzyl, the tertiary butyl group here, selective tertiary butyl, and then we coupled it with the compound what we got before with the caprolactam synthesized and universal deprotection. We got the balanol compound. So as we discussed, this compound is designed for IspE inhibitor, which is designed by the docking method, and we tested in the IspE. It's not active. So we changed it to IspD, and it was active against IspD with this one. And IC50 was 5 micromole of this compound, and that too, it is against the Burkholderia. The Burkholderia is a selective agent, and is a biothreat reagent. So it is a very good successful hit compound for us. And again, in the MEP pathway, after the MEP CDPME, the next is the CDPME2P, and this compound is completely unavailable. So we synthesized for the first time this compound, and since the shortage of time, I'm not going to talk about the whole synthesis steps, but finally we made the compound, and we have the kinetic analysis, and we are screening compounds for this, using this as a substrate for the IspF inhibitors. Similarly, the MECPP, the cyclic compound, also we synthesized. These are all like 100% pure chiral pure compounds we synthesized in our lab, and this one will be used for the screening too. In the other side, we also try to understand the mechanism here. So if you see here, this is the MEP, and we know IspE is necessary to make the CDPME2P, CDPME. But here, if you see, using the IspD, we made the compound, a different the compound which is necessary for the IspF for the cyclization. So this is completely by the different route. So what we are trying to tell here is, if you remember in the first few slides, I told you for the E. coli, IspF is not there, and how it is able to complete the synthesis of the final compound. But here, this mechanism explains it doesn't need a IspF. With the IspD itself, also you can make the compound, necessary compound for the synthesis of the MEP, the diphosphate compound. So this is also, and we also do a lot of mechanism studies and trying to see, explain how the drug resistance and all those things are happening. And so far I have explained about the MEP pathway and enantiomeric synthesis of the MEP, CDPME, CDPME2P, MECPP, and assay determinations for this one. And we also discussed about the discovery of the lead compound against both IspD and IspF, and try to explain the new mechanism, how the bacteria, the how the E. coli is able to complete the MEP pathway. And finally, I would like to acknowledge my collaborators from Colorado State, UNMC, India, and my postdocs, Xiong and Benson, undergraduates, Harry Smith, Iran, and graduate students, Hangjin and Joel Fredson, my technologist, Amanda Bob, and finally the funding from NIH, NRI, and UNMC, and VA. Yeah, thank you. And let me know if you have any questions.